All right. Thank you very much for inviting me to speak to everyone today, and thank you for tuning in. I'll just state up front, I'm speaking to you as a scientist at the U.S. Environmental Protection Agency. Everything I'll tell you is my opinion. That doesn't mean that the EPA told you. It just means that I told you this. My slides are cleared, so if you would like a copy of them, just send me an email and I can send that to you. And yeah, with that, I think I'll just jump into things. Let me make sure I'm showing my slides. Let's see.
Yeah, you're showing your slides fine.
Great, perfect. All right then. So I work at the Office of Research and Development. I know you heard from Rusty Thomas last month. He's my boss, many levels above me, but I just wanted to give a quick summary. Office of Research and Development is the scientific arm of the U.S. Environmental Protection Agency. In 2018, we published over 500 peer-reviewed journal articles, and the research is conducted out of 4 national centers. I'm in the Center for Computational Toxicology and Exposure, but there's also a Center for Public Health and Environmental Assessment, Environmental Measurement and Modeling, and Environmental Solutions and Emergency Response. And the picture I'm showing you is our facility in Research Triangle Park, North Carolina, which I hasn't physically been into since March, thanks to the pandemic, but we do actually have 13 facilities across the United States. This is just the largest one. At that facility, there's more than 2,000 researchers, and the research is done both by federal scientists like me, but also contract researchers, postdoctoral, graduate student, postbaccalaureate trainees, and then there's uniformed members of the U.S. Public Health Service as well, which they actually end up getting, many times they're doing work just like me at a desk, and then they may get deployed to a hurricane situation or other situations where public health expertise is needed. So the problem broadly that our center, the Computational Toxicology and Exposure Center, is focused on is really related to how chemicals are related in the United States. So here's a cover from New Scientist from back in 2014. And you can argue all day about the numbers if you want, but they capture the problem beautifully. We've made 150,000 new chemicals. We touch them, we wear them, we eat them, but which one should we worry about? And they show a shirt, piece of cosmetics, lipstick, and an apple that you might eat. And certainly, being from EPA, I have always kind of thought more about the things like the apples, what's been bioaccumulated in it, what's been sprayed on it. But actually, any of these are a source of chemical exposure to people. And if you look in blood, so this is a study done by Dean Jones down at Emory back in 2012. They found signatures of at least 3,000 chemicals in blood. Now that's not a surprise. Blood is made out of chemicals, but many hundreds, if not more, of those chemicals had evidence that they were actually exogenous. So that again, we have to eat food, we have to do various things, but still a man-made chemical. And so chemicals in the United States are regulated by a tapestry of laws. There's no single U.S. Chemicals Act or anything like that. There's chemical testing and safety depends on really how it's intended to be used. If it's a pharmaceutical, then it's covered by the FDA. There's various levels of human testing to prove efficacy and safety. If it's a pesticide, there's laws that say basically that you need to, if it's intended to kill something, you need to use animal testing to show that it doesn't kill other things and doesn't have unintended consequences. And then many other chemicals are covered by a law in the US known as TSCA, which is the Toxic Substances Control Act. And in that one, it's more of a innocent until proven guilty sort of approach of you look at the chemical structure, and if it seems reasonable and you're not seeing any other evidence, you try to make a decision based on that. And so the level of testing that we have could be everything from a drug that's had human clinical trials to a chemical that has only ever been examined from the level of structure. And so right now there's about 10,000 chemicals in commerce that are produced or imported in the United States and potentially in the environment. And so even if we wanted to, and the law said we could, you just couldn't test all 10,000 of those chemicals with traditional technologies for hazard assessment. That would be animal studies, exposure studies, human clinical. That's just not going to happen for 10,000 chemicals. So There's been a great deal of interest in what are now termed new approach methodologies, or NAMs. And that includes everything from high throughput screening, in vitro studies to assess bioactivity, such as the Tox21, ToxCast projects that have been developed over the past decade, to high throughput exposure estimates, where if you give me a chemical, can we anticipate how it might be used? And based on how it's used, can we say how much of that chemical we expect you to get exposed to? And then high throughut toxicokinetics, which I'll go into more detail here, but really linking that dose, that exposure dose that you might receive to in vitro bioactivity. And you can use this information, and we have kind of this cartoon version of this plot over on the right-hand side of this figure here. You can use this information to make risk-like prioritizations. And I'm certain we could pick anyone on this call right now, including myself, and ask for all sorts of reasons that any of those three types of data or predictions I just gave you would have uncertainty. But what we can do is, if we carefully quantitate that uncertainty, then we have a distribution. A potential hazard is represented by some sort of curve. Potential exposure for one chemical is, again, some sort of distribution. And we don't necessarily know exactly where the value is within that distribution, but it might have a central tendency. And then we can check, and if you have 10,000 chemicals, you're going to be much more interested in a chemical where it's possible that those two distributions overlap than you are where a chemical, which may be uncertain, but there's no possibility of overlap, at least for the average person. And so all of these tools, if the uncertainty is carefully quantitated, can allow us to do some sort of prioritization and identify chemicals that would be much more worth pursuing research on. And all of this relies on in vitro and vivo extrapolation. And that's really the use of in vitro data to try to predict the phenomenon that we see in vivo. And so this is best illustrated by the parallelogram approach, which I keep digging, and the farthest back I've gotten to is Sobel's 1982, referring to the parallelogram approach. But the basic idea is the traditional tox testing is up at the top side of this parallelogram, where you had rodent in vivo data and you somehow tried to extrapolate to humans with rodent in vivo data. And I've come to toxicology recently enough that seems as absurd to me, and not negatively absurd, but as challenging to me as in vitro to in vivo extrapolation. The basic idea is you're trying to go from one species to another. Well, you could also do that in the test tube. You could do that in glass. So you could look at rodent in vitro data and try to extrapolate to human in vitro data and maybe learn something about the differences between humans and animals. But you also, what we're in the 21st century with things like ToxCast and Tox21, we're trying to go from human in vitro data to human in vivo data. But we, again, like I said, I don't think that's a perfect thing. That seems to me as hard as going from an animal to a human. We're going from a test tube to a human. But a place you can test that is actually on this final arm of this parallelogram, go from rodent in vitro data to rodent in vivo data, where we have it, and start to see how these things work. And that's actually what we do in a lot of toxicity testing, and that's what we do something in toxicokinetics. So this in vitro and vivo extrapolation problem of going from the test tube to the real world can be broken down into two parts. And the first part is what I'm really going to focus on. And I'm sorry, it's broken down into two components. I'm sorry, my finger overwrote that there a little bit. The first is is the toxicokinetic portion, so that's absorption, distribution, metabolism, and excretion by the body. And we can do that using an empirical PK model or even a physiologically based model like we'll focus on here. Then there's also a component on toxicodynamics. My liver is not someone else's liver. My genes are not someone else's genes. There are differences from individual to individual in the dynamics of how my body processes a chemical. And as important as that is, and that is very, very important, the high throughput tools that we have right now don't address that as much as we would like. So the issue with in vitro and vivo extrapolation is that you want to use a pharmacokinetic model to do that, or toxicokinetic model, but we simply don't have that information, at least from traditional sources, for most chemicals. And so this is work done by my colleague Barbara Wetmore. And in the first phase of ToxCast, they're looking at chemicals and mostly pesticides. Second phase, they're looking more at industrial chemicals. And in both cases, there's only 5 to 10% of the chemicals where they could generate this in vitro toxicity screening data actually had any sort of pharmacokinetic data. So what Barbara Wetmore and Rusty Thomas started working on is using in vitro tools developed originally by the pharmaceutical industry to try to bridge that gap. And so we can think of these now as new approach methodologies or NAMs for toxicokinetics. So we really need toxicokinetic data for numbers comparable with what high throughput screening for toxicity is doing. And we want to generate this. And so these technologies were pioneered by the pharmaceutical industry, who wanted to be able to prospectively evaluate how effective a clinical trial might be before you actually go and conduct a clinical trial. And so they're in vitro tools that are allowed to allow you to assess toxicokinetics. And so our primary goal was to provide a human dose context for in vitro high throughput screening data. So IVIVE, in vitro in vivo extrapolation. A secondary goal, and I think this is why I'm talking to this particular community today, was really to make sure that these tools were based on open data, open models for evaluation and use by the broader scientific community. So just real fast, what sort of in vitro data are we talking about? Well, there's really just two measurements that we focus on. One is an overall rate of a parent compound. So, bisphenol A is glucuronidated to bisphenol A glucuronidate. We don't assay bisphenol A glucuronidate. We would just look in vitro, incubate bisphenol A with hepatocytes, and look over time to see if that compound decreases, if the concentration decreases, and estimate a rate of clearance of the parent compound by the hepatocytes. We also use rapid equilibrium dialysis well, or there are other ways of performing this measurement, but this has been the mainstay for our measurements. It's a double well with separated by a membrane, and that allows us to estimate how much of the chemical binds to plasma protein. And that turns out to be very predictive, not only of things directly related to plasma protein binding, but also of partitioning throughout the rest of the body. And so over the last decade, we have collected data on 800 chemicals, mostly non-pharmaceuticals, mostly these environmental plasticizers, industrial compounds, consumer product chemicals, and built out this space. And so this is higher throughput than traditional toxicokinetics, but this is not quite the speed of like the Tox21 project, which can test 8,000 chemicals in a day or two if you can design an assay that fits that platform. So defining exactly what I mean by high throughput toxicokinetics, it is not just the in vitro data. The in vitro toxicokinetic data are incredibly important. We can't do it without them. So here's a series of papers that we've drawn from. But we also need a model that can be parameterized with that in vitro data. And that's mostly what I'm focusing on talking to you about today. And so we've been developing EPA's generic toxicokinetic model over the past 5 years in a series of publications. But I don't, so first I just want to emphasize, it's these two things together. It's the ability to generate data in vitro and a generic model that can take that in vitro data and make a prediction. Those two things together are high throughput toxicokinetics. And so I could be working on high throughput toxicokinetics, and I could be entirely in the lab making measurements, or I could be entirely sitting in a computer making a better model. I consider both of those to be high throughput toxicokinetics work, and it's really the union of those two together that are the field as it is today. Now, the idea of a generic PBTK model has been out there for a long time. The earliest reference I can find to actually using a general PBTK model actually goes back to this Rusty Thomas scientist who I've been basically interacting with my entire career. At this point, I think I just started college, so he'd already published in Toxi on he was doing Monte Carlo with the generic PBTK model, but he's not the only one. About Slobe has papers on Mackay. Basically, there have been people studying the idea of generalizing the approach to a PBTK model for a long time, and perhaps most famously, the SimSip group out of Saqqara now has developed a generic population-based model. So why the heck did we at EPA build another one? I've just showed you a whole bunch of them. Well, in 2000, remember we started this in 2015. At the time, there was SimSip and there was ADMET Predictor GastroPlus from Simulations Plus, really the two big guys in the field. And they were really focused on drug leads, where you'd spend a lot of resources because you thought you prospectively might do a clinical trial on this. Our needs at that time were very different. We wanted to screen 1,000 chemicals, and we wanted to do it with less than ideal data. We don't go, if this was my billion-dollar drug lead and I'm at a pharma company and the analytic chemist says, oh, I can't quite develop a method for it, you would go back and spend another day developing a method. If it's plasticizer number 22 and we don't quite have a method, we just move on to plasticizer number 23. So we had a different emphasis. Other models that were available at that time were Indus-Chemfate, but that was really focused on Michaelis-Menten metabolism of volatile chemicals, and most of the ToxGas library is deliberately non-volatile. And then there's a tool called PKSim, which at the time, if it had been in R, we would, which is a statistical language, we might have just used that. It didn't get that until 2017, and so now it seems kind of foolish that we have these two approaches, but we do have two separate approaches. Now, PKSim is much more focused on bespoke models that are designed for specific chemicals, but still in the wash, you know, HTTK is its own separate thing. Now, HTTK does not have a GUI interface, a graphical interface, but both the Plethum tool, which is just published this year, and WebICE from Nicetum, that's the US, oh, I can't come up with the whole acronym, but they are part of the national, well, they serve 12 different agencies in the United States, including the National Toxicology Program. They both provide GUIs to use HTTK and other models, as well as pre-computed values are available from the CompTox dashboard. So regardless of the wisdom, we went ahead and built a generic physiologically based toxicokinetic model. And in that model, some compartments are very simple. You know, so blood is a well-mixed unit. I mean, we do separate it into venous and arterial blood, but basically it's a box that is all at one concentration. Other compartments are separated into the tissue and the blood, like the kidney here, kidney tissue, and then there's the kidney blood. And then we focus really on the gut, the liver, and the kidney in our primary model, and everything else gets lumped into something we call the rest of body. So clearance from this model depends on two parameters. One is the metabolism estimated in the liver, and we get that from the in vitro clearance and binding estimates. So that's directly where we use that. Then there's excretion by glomerular filtration in the kidney, and that we really just use the protein binding in vitro data. And so the model parameters for this are either physiological, are we describing a rat, a monkey, a human, a dog, and that they can actually be varied using Monte Carlo simulation. And there's a whole paper on, and this is not the first on this one either, on by Carolyn Ring is the first author, and as much as I'd love to, that would be basically a whole half hour or longer webinar, and maybe you can have Dr. Ring give that at some point. But variability is simulated physiologically, but then there's these chemical specific parameters, and so there's physical chemical properties like log P. We tend to predict those from structure. We prefer the Opera tool, which is another open QSAR tool. You need partition coefficients, you need the in vitro measured plasma binding and metabolism rates, and maybe you can predict those from structure. That's a different area of research right now. All right, so we need to get tools for chemical specific PBTK parameters. And again, this is not something where we're the first ones to recognize this. So here's a paper back in 1997 looking at PCBs, trying to get partition coefficients and generalize across that. And then over time, Poulin and Thiel particularly have pushed new tools for this. There's a Walter Schmidt model that we tend to like to use, and a unified model, Pere, Poulin, and Krishnan. So this has been an area of research as well. We're not innovating on that idea, but we have made our own model of this. And basically we took the two, at the time, more recent models, the Schmidt 2008 and the Pere 2010. And then we did something a little strange with it. Those models are based on basic principles of how things bind in the environment, how they distribute in the body. And we generated a database and said, okay, well, how well do they actually do? And so this work was led up by Robert Pierce and published back in 2017. And so we actually calibrated the mechanistic predictions to the in vivo data where we had them. And so we saw things like, so you have the dashed line in these figures for each tissue would be the perfect prediction line, and then things tend to do pretty well. These two outer lines, that's the kind of the confidence interval, and so you can see, you know, for things like muscle that are fairly homogeneous, or even skin, we do very precise predictions. And if you get over to something like the brain, where there's the blood-brain barrier, which we can't model transport right now, so we do a worse job. And so this actually gives us confidence or lack of confidence in these predictions. And so we then evaluated all of this by trying to predict the volume of distribution for 500 chemicals from a paper published by Wambaugh in 2008. And the root mean squared error was, about half, which is for us, that on a log 10 scale translates to a factor of 3. And that's exactly where we want to be for trying to predict things from in vitro measurements. So our tool is publicly available. We have an R package that we cleverly named HTTK. And it is available on CRAN. It's also available on GitHub. If you have R Studio, so R is a free statistical language. Anyone on Earth can install R for free. Once you have that installed, you can install HTTK for free. And then you have all of our data sets. You have all of our models. You actually have what are called vignettes, which are walkthroughs of actually how we've made the figures in various papers. And then there's a reference manual, which is not fun reading, but is at this point 176 pages long, describing how to use all the functions in there. And so right now there are 987 chemicals with human measured data. There's another almost 200 with rat. And this tool allows you to do in vitro in vivo extrapolation. And it was described in a paper in Journal of Statistical Software. So why did we do this? This just gets to the whole point of the Open Talks meeting. There's a nice paper with Eva McClanahan as the first author back in 2012 that although the publication of a PBPK model in a journal is a good step, it's hardly the last step. And often there's a lot of difficulty reproducing using a model even once it's been published. And in the US federal government, we actually had an executive order back in 2013 that were really the default state of the new government information should be open and machine readable tools. And so we really want to have models that are open source, the data are open, the models are open, and available to the public to review. And so there's been a lot of thought on how do you verify a PBPK model, and there's a nice paper from Leona Clark back in 2004, where you basically just, you have to assess, why did you build the model? Do we believe how plausible it is biologically? Do we think the math is right? Did we get even if we got the math right, did we get it into the code correctly? How well does the model work? And then assess any specialized analysis. And as I stumble over these words, just telling them to you, you can probably believe this is not the easiest thing to do. You can spend a long time doing this. And so what I'm going to wrap up with is just basically arguing to you that HTTK and generic modeling actually allows you to do a lot of this much more easily than we could in the past. So if I have a chemical specific model, I made my model for bisphenol A or whatever, and I want to assess how well it does, I think the best tool to use, I was trained by Woody Setzer also at EPA, is to take your observations and look at how well they correspond with the predictions. In a perfect world, all your points would be right along the prediction line, or the perfect predictor line, the dashed line, and I'd never believe you that if it was that perfect. But if there's a little bit of scatter, that scatter gives you an uncertainty. You can look for bias. Do I overpredict? Do I underpredict? And you can consider whether or not using that model to extrapolate. The problem we have, again, at the Comptox Center, if we've got 10,000 chemicals to look at, is that we cannot build that model. We don't have observed concentrations for thousands of chemicals. So we don't have the data. So what we want to do is parameterize a generic model and see how well that generic model does for what we're doing. So you might say that seems ridiculous, and I'm showing you here for chemical X, the chemical specific model has a lot less scatter, so it has less uncertainty. Right now, my model doesn't seem to have any bias, but what's the advantage of a generic model? Well, the trick is I don't have to just use my generic model on chemical X. I can also use it on chemical Y. And I can start to look at how well do I perform across many chemicals, and then I can look at chemical Z, and chemical Z, I find that I always overpredict. So there's bias for chemical Z, and then I can start to look at the domain of my generic model. What is it? What features of chemical Z lead me to think that I tend to overpredict for chemical Z? And so in this case, by having a generic model, for any one chemical, it doesn't do a great job, but for every in vivo chemical that we have data on, or every chemical that we have in vivo data on, I can run my model. I can start to get an idea of how it's going to do for chemicals where we don't have data. To do that, I really need some computational resources. And so one thing I'm very excited about is just publicly available this year is what we call the CVT Database. It's a public concentration versus time database. It's developed by Riza Sayre and Chris Grolke at the EPA. The published version right now has 198 analytes drawn from over 500 studies. routes include intravenous, dermal, oral, and it's mouse, rat, human. And so it gives us a database that we can start to compare our high throughput toxicokinetic models and see how well they do. One feature of the pandemic, I feel like Microsoft here, it's not a bug, it's a feature. Our building is closed down. A lot of the great pharmacokinetic researchers that we have at EPA are trying to do lab work from home. And depending on what they have in their basement, that may not be possible. So we've actually had ongoing curation by a lot of the experts in pharmacokinetic experimental work. They're still going over the literature, digitizing data, and adding that data into the concentration versus time database. So that's happening right now. And I'm extremely grateful to those people because they're continuing to push the data that we have. We are also currently hiring a position for a database engineer to kind of supervise this whole database. And that's available on Zintellect right now. It's at a master's level. And I think that closes next month. So there's plenty of time to look into that. And so this is an ongoing effort. So what can we do with this? So we have the HTTK package. It allows you to predict tissue concentrations depending on oral, intravenous, or gas exposures. It allows you for in vitro to in vivo extrapolation. And so what we really want to do is see how well these models work. And so we have a standard, current version of HTTK is 2.0.1. Right now, a standard PBK model, but we're working to improve and add different features. So there's a gestational model in development. There's an aerosol exposure model in development. There's a dermal exposure route model in development. And so each of those models is only released once we have a peer-reviewed publication. Then we put out all the code. We've gotten most of the bugs. But if you have, if you're interested, we can actually share information under a legal agreement, but we'd actually have to develop that agreement. We assume that there will always be coding errors and oversimplifications in every model we develop. And so what we really want to do is evaluate how bad the models are. And so a great case study was just published by Matt Lanakis at the U.S. Air Force, working with EPA. This is a generic gas inhalation model. So this is a very important route of exposure in occupational settings. And so this paper was just published in Journal of Exposure Science and Environmental Epidemiology. It was based on two general inhalation models that were previously published, the John Gamillion models known as Indus Chempate, and one by Harvey Kluhl. And what's different about this model is it's designed to be parameterized with the HTTK data. So out of the box, this inhalation model works for over 900 chemicals in human and 180 chemicals in rat. Now, not all of those chemicals are necessarily ever going to exist in the gaseous form, but that's the beauty of a generic model. We can start to evaluate this. And so what we did is we compared the predictions of this inhalation model with data from the CVT database. So we looked at inhalation data in humans and rats and what we predicted. And we were able to get the bias, and there's definitely some bias, but we're actually able to quantitate what the root mean squared error is and say, yeah, we're within a factor of 3 on a lot of this stuff. Now, what was really interesting is, okay, well, one thing I should say is, the first time we did this, of course, our model looked like crap. Having the data there helped us catch errors in the model development that we wouldn't have otherwise ever seen. And having the data for lots of chemicals helped us catch bias of, oh, we seem to be handling log P wrong, or we seem to be handling other features wrong. One thing I hadn't anticipated prior to starting this project is this also helped with data curation. When we started making plots, there was one chemical, and I'm blanking on the chemical. It's one of the great, it was one of the landmark chemicals, volatile chemicals of the 1980s. And we didn't have one data set we had wrong. And I just remember saying to them, well, boy, nobody's going to believe this if we don't have this chemical correct. And I can't come up with which one it was. But the points are like 4 orders of magnitude off our curve. And what happened as we looked into it is actually the units in the caption of the figure of that paper were wrong. In the text of the paper, it said they were 1 unit, and in the figure, it said something else, and the figure caption was wrong. And we were able to catch that just because those points were such extreme outliers. So having a generic model also helps us with the data curation. And so what we're able to see is that with this gas model, we get an R-squared of 0.67 for predicting C max, and we get a R-squared of almost 0.8 for predicting AUC, time under the curve. All right, so we have this generic model. Here's the criteria from Clark et al. 2014. So what's the model purpose? We want to do rapid in vitro and vivo extrapolation. We assess the structure by using a diverse chemical library, basically every chemical that we can find. We make sure that the math descriptions make sense by arguing with peer reviewers. It's just how everybody does it through journal articles. But we assess the computer implementation by actually making the code available on GitHub, on CRAN, and believe me, people find bugs, and it's extremely valuable. People will point out what we've done wrong. Usually we fix one bug and that introduces a new bug, but we make the code available and it evolves over time. We have this concentration versus time database now to assess model fitness, and then specialized analyses like the HTTK POP simulator have been published separately, and then that's actually being revised right now by Miyuki Breen, a postdoc I'm working with at EPA, who's updating the population simulator. So in conclusion, high-throughput toxicokinetics is designed to allow us to adjust high-throughput screening activity data into an in vivo system, and we want to do it in an open source, free, and evaluated way. We want to characterize any model bias and establish the uncertainty in that model. And that really, in this case, quantified uncertainty is confidence. If I know it's good within a factor of 2, I'm a certain level of confidence that's greater than if it's only good within a factor of 10. By using a generic PBTK model, that actually makes a whole lot of the verification easier because we can just run it for lots of chemicals and see how it works, as long as we have this awesome concentration versus time database that Riza Sayre, Chris Wilkie developed, and now a team of scientists at EPA is continuing to populate. So I hope it comes across that there's been a ton of people working on this. The HTTK team has a variety of ongoing projects right now. I've had a lot of support from my management, both in Rusty Thomas and Mike DeVito at EPA, and then a long-time collaboration with Barbara Wetmore, who now runs a lab at EPA. But we do have a contract with Ciprotex. They've been generating data on hundreds and hundreds of chemicals for us, and then very productive relationships with a lot of postdocs and other researchers and collaborators outside the agency. All the references are available here. Like I said, these slides are cleared, and I can send them out to you. And hopefully I left a little bit of time for questions. I'm happy to answer anything you want to know, well, that I know.